Okay, so let's go for round two of PTM. So next up is George Rosenberger. So I guess he's been around all week, so maybe some of you have spoken to him, but George was a PhD student in the group here in Zurich and was a key person in the development of OpenSWAT. And now he's a postdoc now at Columbia, but we're still managing to convince him to occasionally work on the software. And in the last part of his PhD, he was very focused on software to kind of do basically what Tina was describing, but in a formalized and automated way. So that's this inference of peptidoforms. So we're looking forward to hear about that. I guess you need this.
Great. Thank you very much, Ben, for the kind introduction and especially for the invitation. It's a pleasure to be here. So we learned a lot from Tina's talk about the complexity of measuring PTMs by mass spectrometry, all the difficult experimental steps. And now first the bad news, it's just as bad on the computational side. The good news is that more than a decade of research has produced nice solutions that actually enable to do automatic site localization and confident detection of PTMs at very high throughput for the easy to medium difficult cases. And in my talk today, I will speak about the similarities and differences between DDA, SRM, and DI and the computational considerations, and I will specifically focus on the improvements that we can gain when using DIA. So I will briefly start with an introduction about the general concepts. I will talk about this inference of peptidoforms scoring system that we developed particularly for DIA. Talk about a practical application where we try to showcase what kind of benefits we can gain by using this strategy. We'll briefly talk about the special case of assessing co-eluting peptidoforms and some recent developments in this regard, and I will finish with conclusions. So let's start with the introduction. When we talk about PTMs to others as proteomycists, usually we think about PTMs like phosphorylation or ubiquitination. These are part of very complex biological systems like signaling networks. We commonly can detect 10,000 of phosphopeptides within a simple biological system, and all of these, they interact together, there's crosstalk, all of it is difficult, right? But what's very fascinating to me is that sometimes not everything is complicated in biology. Sometimes a little thing can make a very big difference. And that's an example I will showcase a few times today. This is an example that is relevant for clinical applications in cardiovascular disease. So this is a representation of an arterial artery on the side. We have here the arterial walls, and what's happening here, especially in terms of cholesterol, is that cholesterol regulation is very important to maintain healthy arterial walls. So there's a large particle in blood. It's called high-density lipoprotein. It consists of about 100 different proteins and different lipids. And in interaction with macrophages and other systems, it can mediate reverse cholesterol transport and ensure that the cholesterol levels here are tightly regulated, thus inducing antithrombotic effects. Now, it was recently found that in people affected by cardiovascular disease, this HDL particle is rendered dysfunctional, and this is caused by a single oxidative modification on the major component apolipoprotein A1. So single oxidation can be very important for the clinical picture that we have when people have cardiovascular disease. Now, oxidation is a very difficult PTM to measure. It can be both a biological part, but it can also be a technical artifact. And this is really the goal for our software and our workflows. We need to be able to differentiate for this between technical and biological components of the signal that we are measuring. So that's the benchmark for our tools. And now we'll have a brief look at the considerations we need to do when analyzing mass spectrometric data. Okay, so the first part is peptide queries, sensitivity versus selectivity. So you know probably all how this works in DDA database search, the considerations you have to do. So you set up your workflow, you specify a FASTA sequence database, you specify the PTM types you're interested in, and the search engine will do peptide spectrum matching. Now, from Tina's talk, we learned that in targeted proteomics or in DIA, we need to specify peptide query parameters to detect accordingly the peptides, and these need to be specific for what we're targeting for. And these peptide query parameters, as you learned this week, they consist of N transitions, precursor and product mass overcharge, the normalized retention time, and the fragment ion intensities. Now, the question here is always, how big does this N have to be? And the other important question when we're designing mass spectrometric methods is, do we have enough transitions to choose from? And as part of the original SWOS MS paper by Ludovic Géé, and later in an algorithm by Hannes Röst called SRM Collider, they investigated this question from a theoretical point of view. And the main question was, do we find enough transitions that are unique so we can achieve specificity for detecting peptides? What they found is that with the original SWOS MS precursor isolation window, there are at least 5 transitions for more than 90% of the peptides. So this is great. We can use only 5 transitions and actually be confident that a specific peptide sequence can be detected. The big problem here is when we go to PTM, this is only the case for unmodified sequences. So when we look at PTMs, as you learned before, differential site localization, this will be very difficult, and we need other strategies. So the naive strategy, naive approach is we just increase the sensitivity of our peptide query parameters. And then we will unfortunately have a substantial loss of sensitivity because there are not enough site determining fragment ions for all of the peptides that we're interested in, and usually they're of lower intensity, so we will lose a lot of the signal. For this reason, there are these 2 strategies, closed and open modification search, that have been originally developed for DDA and then propagated to SRM and now DI. So with the closed approach, we actually specified peptide sequence and modification mass and type. In DDA, the algorithm will then only match to those peptide spectra that are actually within the expected range of the modification mass. And then in a second step, we assign the site localization. So the open modification search, we do this a bit differently. We first detect the peptide sequence. We then try to figure out what's the modification mass and type, and then we do site localization. So let's have a look at the close modification search first. I said PSM with small precursor mass, and this is really what most search engines do. In peptide-centric scoring, it's actually an interesting case and something that was developed a bit later on. So first in SRM, people are already targeting for the specific precursor ion, so that's not a big case. But in DIA, we first focused on the MS2 level and only then came back to the MS1 later when the field shifted more towards PTMs. So the 2 general strategies. First, we can include MS1 scoring. This is available in Skyline, SpectraNote, and OpenSWOS. And the second approach is to assess the MS1 signal independently. So either we just say we combine everything and hope for the best, or we first detect the candidate peak group and then we specifically validate that this peak group has a specific precursor mass. One approach here, this is MS1 filtering that's implemented, for example, in Skyline, and maybe I've learned about it this week. It works like this. So first, the MS2 ions are extracted from the MS1 scans, and then with the parallel MS1 scans, the precursor ion signals are extracted, and just by visualizing this, you can confirm that actually this peak group here on transition level has this very specific precursor ion mass overcharge. And this gives us additional specificity because sometimes we're only interested in the peptide sequence and we don't care whether it's modified or not because we want to do protein inference quantitatively, but sometimes that can be very useful, especially in our case with post-translational modifications. Second part, open modification search. Also, several others have talked about this. Alexi, for example, with his tool MS-Fragger. So here, first, the PSMs are computed with a wide precursor mass tolerance, like several hundred Daltons. And because of the recent developments in instrument performance, with very high resolution, we can only use a few partial unmodified fragment ions to have an accurate match, and then we can estimate the delta mass from the precursor and the fragment ions. And there are many tools that support this. For DI specifically, that's MSplit DI, which was also mentioned a few times this week. This is an illustration from the MS-Fragger paper, and it kind of illustrates the vision that there is for open modification search. So in closed modification search, you always have to put in a lot of prior knowledge. You need to define the residues, and you need to define the post-translational modification types that you're interested in. But with open modification search, the dream is that all of this can be done automatically. You only specify your FASTA database. And these figures here, they illustrate different mass ranges here. Here, for example, we have, of course, the most popular phosphorylation. You can see that this mass is always detected within a very narrow range. Now, what's also very cool with this kind of search, we can learn the residue distributions, the frequencies that these post-translational modifications modify specific sites on our peptides. And that can be very useful to detect new things, and it can also be very useful to detect things that are unexpected to you and that you did not consider in your initial experimental design. Okay, we can also do this in peptide-centric scoring. The most popular implementation being SWAS-Prophet PTM. Tina already mentioned this. We can query unmodified peptide query parameters in other precursor isolation windows and then estimate the delta mass from the precursor and the fragment ions. So this is an approach to generate a hypothesis to detect and identify new peptides with new post-translational modifications. Okay, so we have these two approaches to figure out what kind of PTMs we have, or to validate that we actually have the right PTM type targeted. And now the second component of the problem is the modification site localization. And again, there are two very different approaches. The first is unrestricted search space. So we have here a peptide sequence, and we have a modification type. We know the type, but we don't know where it is. So what we could do is we could just assess whether any of these residues are actually post-translationally modified. The problem here is we have low sensitivity because we always have incomplete fragmentation with all of the methods that are currently in use. The alternative most commonly used is the restricted search space. That's what you usually do in DDA database search, or also, of course, with DIA. We only assess the canonical residues, for example, STY for phosphorylation, and this actually, you can see this right from this picture, this will make our problem much easier to differentiate between these two than between these two. But again, this only applies to non-canonical residues. And there have been recent publications where, for example, non-canonical phosphorylation has been investigated, and that's also something very interesting that you will miss with this approach. But there are always these trade-offs that we need to take. Okay, so algorithmic approaches, that's actually was the main focus of in terms of PTMs over the past decades, starting from DDA. There have been algorithms like A-score or Andromeda PTM score, and to take a probabilistic approach. So what they do is they usually employ a binomial model, and they assess how likely do you expect a specific site localization in comparison to the other alternative explanations. There are also other implementations like the Mascot Delta score, where just the search engine scores are compared, and they need to be reasonably different between two positional isomers that you actually trust a particular site localization. And then more recently, target decoy approaches like Lucifor have appeared that actually enable you to estimate a false localization rate. So these scoring systems, they usually give you a probability, like, okay, we have a total probability of 1, and then this is assigned across a different site localization, and here you can have actually a real error rate model. Okay, the last consideration we need to take is the informativeness of fragment ions or transitions. So Tina has showed a lot of examples where single transitions need to be informative and you make decision on them. And this actually varies a lot between different systems. So it varies in terms of specificity for the numbers of matching peptide isoforms. For example, with phosphorylation, it depends on the STY residues that you have in your peptide sequence. Then it also depends on the fragment ion length. That's also a crucial part. And particularly, it is reduced for some modification types, for example, neutral loss of phosphorylation, which Tina also explained. Okay, then when we look at this problem and we want to design a system that does this for us automatically, eventually we'll come to the challenge to assign confidence to individual transitions. And that's very challenging with single fragment ions in spectra, so we believe transitions are more informative. This here is an example. We have here 3 alternatives, and they're closely related, and this is the fragment ion spectra. And what you can see here is that there are many shared transitions. All the black ones are shared between all of the 3, and the double-colored ones are shared between 2 of the different positional isomers. And there are also fragment ions that are unique. But for example, for the green isomer, we don't have any unique transitions. So what we need to do here is we need to make inference. We need to infer from, if we detect the blue and the green and the pink and the green, it's more likely this isomer than the other 2. And that's very important for us to consider in our scoring system, that we can deal with ambiguity, with evidence that counts only partially, and we need to find a way to integrate all of this. So just to summarize quickly, challenges, large precursor isolation windows, Tina mentioned this, but we can actually deal with this properly if we have a decent MS1 signal. And then core fragmentation of closely related peptide forms, and this makes site localization difficult. So I will now move over to the opportunities of DIA, continuous fragment ion spectra, and we can make use of this. And eventually our goal here is to improve the quantitative consistency. So I also show here the picture that Tina already showed. Actually, this is from the protein that I showed in the introduction. This is an apolipoprotein A1 peptide, and it's present in several different peptide forms. So it's different modification types and different site localization. That's actually a clinical sample. This is blood plasma from patients that were affected by cardiovascular disease. Some of those even died of stroke or other complications. And what you can see here is that these different oxidations and other modifications actually are very difficult for us with the existing technologies to assess. And thus it's crucial that we develop new approaches. Okay, so let's talk about IPF. Realizing in the middle of the presentation, and I never explained what a peptide form is. So peptide form is just a peptide sequence with specified modification types and specified site localization. So it can be a positional phosphopeptide isomer, but in general, it's just closely related peptides that have a very specific molecular composition analogously to proteoform. Okay, so let's start with the motivation and vision for IPF. This started from the OpenSWOS workflow. The ultimate goal of the OpenSWOS workflow is to take DIA data and just generate a quantitative matrix. We have peptides on one dimension, and we have different samples and conditions on the other. And all of the individual values that are in here, they have 2 important metrics. First, it's confidence that it's actually a specific peptide, and the second is a quantitative value. Now, if we want to achieve the same thing for modified peptides, it's going to be much more difficult with the established approach and concepts. So, for example, if we do this in a label-free quantification workflow used for DDA, what the tools usually do is first they go in one direction. So within each one, independently, we assess peptide spectrum matches. We have a confidence for the match, and it's filtered for this confidence. For example, 1% PSM FDR. And then on top, we do site localization. We again filter this to 1%, maybe false localization rate. And doing this iterative filtering actually reduces a lot of the hits that we have. And what the algorithms usually do is they just say, okay, we do this kind of with a lot of holes, and then we do an alignment, and the alignment is quantitative. And if you have simple samples, that usually works. If you have very complex samples, then the illusion patterns can change, and you will align the wrong things, and you might actually quantify, or you might assign the wrong identification to particular quantifications. So our vision for IPF is to do this on the peptide form level. We have here specific peptides. They have specific site localizations and modification types. And for each of the values, we have a confidence that it's this particular peptide form. So we don't care about whether, or what's the detection confidence. We don't care about the site localization confidence. We want to have this integrated. We want to have an absolute simplification, and this simplification will then enable us to do this over hundreds of runs. That's the main asset. So with IPF, we cannot really improve a lot in this direction, but we completely focus on this dimension because we believe we can achieve this dimension or very well already with the established workflows, but this dimension is lacking, and we believe it's very important for biological applications. Okay, so I will briefly go into the details how this works. We start with an input spectrum coming from DDA or DI-Umpire, and then we select detection transitions. You have learned all about this in the library generation workflow session. We usually select the most intense transitions that follow some patterns because they will enable us to accurately detect potential peptide sequence features within the SWOS-MS map. And then a very important part, which is identical to the DDA spectrum-centric approaches like ASCORE and others, we generate identification transitions. So we need to supply a model of how a particular modification type works. For example, here we have phosphorylation on 2 different residues, and we generate all the theoretical transitions that could differentiate, but that are also shared between those. And we can do this with all combinatorics. If you have 3 different modification types and several different site localizations, this completely explodes. But actually, this problem is not too bad. It will just generate a single list of identification transitions that we then append to the spectral library. Spectral library has 6 detection transitions, but several hundred identification transitions. Okay. So just a few notes here. We always use representative DDA runs, or even better, DI-Umpire pseudospectra. We're here operating on the peptide sequence level. So we need to have a library that is as good as possible in this dimension. And we don't recommend using large repository scale libraries. So if you could generate the biggest phosphopeptide library and you apply it directly to your sample, you might actually perform much worse than what you would do if you use DI-Umpire. Okay, we also recommend RT normalization that improves just as with all other peptide-centric strategies, and you don't need to supply site-localized libraries, but it can be beneficial in some cases. IPF will always doubt what you supplied, and what you supply will only be used to actually find candidate signals. Okay. Then optionally, you can also increase here the selectivity by using site-determining transitions. We don't do this frequently because it will lower our sensitivity, but I will come to special cases where this might be useful. Then you define the peptide query space. Usually you provide a modified Unimod XML that you filter down to the physical-chemical model that you actually believe, including, for example, neutral losses if you're interested in implementing this in the model. And yeah, what IPF also does in this step is an automated generation of decoy transitions on the transition level to estimate the null distribution. This will be important in a minute. Now, the crucial step here is signal processing. This is all done now in the spectral library coming from anywhere, but now we're going to the DIA data, and we assess the data, and we try to figure out what kind of signals can we use. So first is the peak group detection. This is identical to OpenSWOS. We implement this using the OpenSWOS workflow. We go to the data, we extract the detection transitions, we do all of our different scores, and we assess the confidence that this is a proper detection. The second step is then the precursor level, and this is closely related to MS1 filtering in Skyline. We have additional scores implemented in OpenSWOS that extract the fragment ion signal, the precursor ion signal from the MS1 maps and the precursor signal from the unfragmented MS2 maps, similarly to how DI-Umpire does. And then we have scores that check whether actually these ion chromatograms coelute, and then we assess the confidence that these originate from the same peptide. The final level is the identification step, where we use all these identification transitions that we can use for site localization. So we have 6 transitions here, and we extract hundreds of transitions here. And we also compare whether they originate from the same peptide signal. And for each individual transition, we assess the confidence for detection and for this thing. And if you think about it, this is exactly what you do in Skyline when you do a manual analysis. You have your Skyline document, and you have the transitions that you use to detect the candidate signal, and then you extract all of these things here and manually assess this. Now in Skyline, you can do this very well for 20, 30, 50. But if you go to 500 transitions, you need very good eyes and a lot of coffee. And eventually you will get confused, and the algorithm will also sometimes get confused, but it will get confused systematically. So we have some opportunities here to automate very challenging problems. Okay, so how does this scoring work? You learned all about the scores. I'm not going into more detail here. But what you need to know is that we do this instead of 1 level, we do this of 3 levels. It's completely independent. So we have a set of scores for the MS2 peak group level. We integrate this into a discriminant score. We have a target decoy approach, and this gives us an estimate of the confidence for detection of peak groups. We do the exact same on the MS1 level, and we also have here an estimate of the confidence that each peak group has a specific MS1 precursor. Now it's so important to remember that the MS1 signal is usually weaker than the MS2 signal, especially on the triple TOF instruments. This will actually cost us something. If we need precursor information, we will lose identifications and detections. It's a bit better on the Q-Exactive instruments, and I will talk about best practices during the tutorial session. Third level is transition, and you can see here from the histograms, these are many more transitions than we have peak groups. So we also score all of these independently in a huge global model, and we have a confidence for each transition based on this large set of scores that we can all integrate. And that was very difficult to achieve, but it really brings down the benefits that we have in DIA. If you can put confidence on a single transition, this will help you for many different things like PTMs, site localization, SNPs, and all kind of other parts. So that's a useful extension of the framework. The final step is the inference part. We have all of these different components. They're all laid out, nicely arranged. And what we now do is we integrate all of this. So first, we can go to the precursor level if we want to do this, and we will just integrate these scores for each peptide peak group in each running condition, and for each peptide and MS1 signal in each running condition, and we just correct using a simple Bayesian approach. The second part then, when we have this, is we do the site localization using all of the transitions that we have. And there's a simple analogy on how we do this. So let's just ignore the precursor level for the moment. Let's assume we have this precursor confidence, this peak group confidence that you know from the other lectures, and we have all the evidence for the transitions. Now what we can do is we have a few bins. Each bin is for a particular peptide form or a particular site localization, and we have a separate bin that assesses the possibility that actually our signal is wrong and false. What we then do is we go through each transition that we have extracted before. We take the confidence and we see, okay, this belongs to 1 or 2 or 3 of the individual peptide forms, and we break it apart and put it into all of these individual bins. And we do this for all of the several hundred transitions, and at the end we will see where is the most weight, and this is the most confident site localization. The cool thing about the system is we also put all the counter evidence for something particular into the null hypothesis that we actually have a wrong signal. And it turns out that this kind of calibrates the whole system, and at the end what we get is we get a confidence on the peptide form level. So we have the same matrix, but now it's not on the level of individual peptide sequences, but it's really tied to peptide forms. So over several dozen, 200 of runs, we have confidence that this is actually what we are targeting for or not. And that's IPF in a nutshell. Now, of course, the question is how well does it perform? And for this, we have designed a synthetic reference data set. It's based on 500 heavy isotope labeled unpurified synthetic phosphopeptides measured in a complex human cell line background in a 13-step dilution series on a Cyax triple TOF. And although each peptide could be localized at different sites, we only focused on those peptides that were present in a single positional isomer, and we used this as a ground truth. Okay, we didn't do any alignment by trick. This is possible, and the real benefit of the IPF strategy, because it's fully integrated within the OpenSWOS workflow, this can boost your quantitative performance. But we didn't do this here. We only focused on this IPF performance. Also, we did do precursor scoring using MS1 and MS2. We did this for our main application to show that how it performs. But if you do phosphopeptide analysis, you should not do MS1 and MS2 precursor scoring. This will improve your identification detection rate by 10 to 20%. Okay, so here's the benchmark. This figure is easy to interpret, and we compare to OpenSWOS. We have here the 13-step dilution series. In light green, that's the number of OpenSWOS true positives. In dark green, it's the IPF true positives. And in orange, it's the OpenSWOS false positives. And in red, it's the IPF false positives. What you can immediately see is two things. First, we lose a bit of sensitivity. Not all decent OpenSWOS signals are good enough for IPF, but what you can see here is we actually lose most of the false site localization. So we really improve the quality at slightly cost of sensitivity. And what you can see here on the quantitative level, where we have here the dilution steps, and here we have the log intensities in comparison to the most intense run, is that this quantitative performance is actually identical to what we have in the standard peptide-centric scoring approaches. If we look at the receiver operating characteristics, we see that we can achieve about 71% sensitivity at 5% false positive rate. So we're actually at the level that is a bit worse than what we have from the peptide-centric scoring tools, but we have a much more difficult problem to solve, and we believe that this performance actually is good enough for many applications. We also assess the error rate model, and what we found is that the error rate model is actually very accurate. So here this is the estimated FDR by IPF, and this is the true FDR that we have because we actually know the ground truth. And you can see within the useful range, both local and global FDRs, they're very accurate, and this is very important for us because we can now then do good alignment with trick, as you learned in the other lectures. Something to consider here is that when we look at the whole peptide intensity distributions across the dilution series, one important aspect is that With IPF, we need slightly more intense peptide signals than what we can use with OpenSWAS or all the other peptide-centric tools, because we need to be able to identify enough transitions for site localization. Okay, then of course, an important question is, how do we perform against the state of the art? And the state of the art in large-scale experiments is data-dependent acquisition and spectrum-centric scoring and labor-free quantification. So we designed an experiment for this. It's an enriched phosphopeptide sample of a human U2OS cell line. We believe it's representative of a standard phosphoproteomic experiment. We have a treatment, which is nocodazole. It arrests the cells at the mitotic stage, and we have an untreated control. And we have 10 biological replicates for each of these conditions. That's very important. This is our biological replicates, not repeat injections. We measured this on a triple TOF 6600 in DDA and SWOS MS modes. We do a DDA database search and label-free quantification on MS1 level, including alignment with one of the best tools that are out there. And we then apply IPF using the exact same DDA-based spectral library. This is not a saturated library. This is just for comparison reasons we use the exact same thing. And then we do alignment using TRIC. And the goal is to compare the DI versus DDA identification and quantification. That's very important. This is a very focused comparison. This is not. This is really comparing how do we do across a large number of samples and not representative of the full workflow. But what we can see is that if we're actually interested in quantifying peptide form across many samples, we can improve performance substantially. Here on the upper part, we see the IPF detection. We see the IPF quantification. You can see that all the colored parts, this is a confident detection and a quantification event. And you can see that this is relatively consistent. There is some variability. This variability is both combines all of the effects, biological effects, digestion. It includes also, of course, all the MS effects, the LC effects, and the IPF effects too. We have the same here for DDA, and you can see it's a bit of a different picture. We still have good representation for some of the peptides, but not for all of them. We get holes, and this is also true for the quantification. So this is only identification, and this is filtering down everything. But with the quantification, we say just align everything that you can find that has the same precursor. So we completely lose the connection between these two layers. Often this works, but sometimes it's problematic, especially when we scale up the number of samples. So yeah, and we can also kind of quantify this effect here. We have a metric quantified in at least X number of replicates on this axis here. We have here in red the DDA control and nocodazole, and the IPF control and nocodazole, and the number of peptides where we actually have a quantification event. Now, what you can see is we can easily, in this number of samples, quantify peptides in at least 2 of the runs. But if we want more quantitative consistency, what we can see is that this drops substantially using DDA, but it remains more consistent when we use DI. We cannot fix all the biological variability, but at least on the instrument side, we can do a bit better with DIA in this manner. Okay, so how is this useful? That was very important for us to assess, and we found, luckily, a very cool data set to showcase the performance of IPF. So this is based on the work of my colleague Yanxing Liu at the Ebersol lab. Back then, he was working on an interesting project. He was assessing human blood plasma proteomes, and he was specifically interested in can we figure out why blood plasma proteins vary in their abundance in individuals, and can we decipher the heritable and environmental and longitudinal effects. Now, that was a great project because blood plasma is easily accessible, and there are many twin studies that enable sophisticated experimental designs that enable you to actually decompose these effects. So in this study, he had like 72 monozygotic female twins and 44 dizygotic female twins with an average age of 58 years. and they donated blood plasma twice over an average time span of 5 years. These blood plasma samples then were all collected and then processed, and 232 SWOS MS maps were acquired. So in Yancheng's original study, he used this data to study the protein level effect, and we thought, can we also use OpenSWOS and IPF to study the effect of individual peptidoforms, of individually site-localized peptides with specific modification type. And then what we use to assess this is a linear mixed effects model that can actually for each peptidoform independently assess the heritable, the common environmental, the individual environmental, and the longitudinal effects in a quantitative manner. And this is possible because we have the twin study, right? We know how the twins are related to each other, and we know actually, because they donated blood plasma twice, we have this aging effect, and because some are genetically identical versus non-identical, we know, is it an individual or is it a common environmental effect, right? So on the big picture, if we then analyze this data, what we get here, we assessed all these different modification types here, and what we can see that the spectral library based on DDA is relatively comprehensive over all of these samples. And here, what you can see in this lightly gray shaded box here, this is the cumulative peptidoforms that we could detect. So DDA is more comprehensive than DI in this case. So we could not detect everything, but at least a large fraction of this. And then we have large heterogeneity in these samples. Here, this is detection in number of samples. As mentioned, we have 232 and few controls, and not every modification type and peptide is consistently expressed and present in all of these samples. So actually, the median is at about 50 samples. So we have still a large sparsity, but this is due to biological effects, right? And it also varies a lot for different PTM types. Here we have labels spiked in. These, of course, are consistently detected across the full range. Then here on this plot, we have the coefficient of variation. We see this for different modification types. And here we have technical and whole process replicates. So what we can see is that here the biological variation is actually in there. It's higher than the other effects. And this means we should actually find a signal that we can interpret. I will only focus on the example here that we had in the introduction on HDL, apolipoprotein A1, and oxidative modification. So, and as mentioned, it's known that a single oxidative modification here can make a big difference. This is due to the enzyme myeloperoxidase binding to the surface on the protein structure, which induces oxidative modifications on the opposite side of this protein. Now, in our study, we have a healthy cohort of females, but they're at the critical age of their life. And the idea here is that as they get older, there is an effect of this myeloperoxidase. And what we can see is that actually the longitudinal or aging effect here has one of the biggest contributions. There's also a big individual component and the heritable component, but aging seems to be an important aspect here. What's very important with IPF, this is the most challenging site local or positional phosphopeptide isomer, but we also have methionine oxidation contained in the same sample. Methionine oxidation on the same peptide sequence is a technical artifact. It originates from the storage of the samples, and because it's a time series, this goes into the opposite direction than the tryptophan, which is a biological effect. So deciphering these two different effects is very difficult, and with IPF this is possible, and we can use the quantitative results to do biological interpretation. This is only one example. We have the exact same statistics for over 4,500 peptidoforms, and we can really decipher into heritable, individual, common, and longitudinal effect. And we show many interesting applications in the paper if you're interested in this kind of research. So for us, this is one of the key applications of IPF. It's quantitative consistency across many different samples, and we believe that this is an interesting new venture for PTM applications in mass spectrometry-based proteomics. We'll now go into a bit different direction, assessing coeluting peptide forms. This is kind of a spin-off. It's an interesting case because there's a lot of interest in this research now. You have also shown some results from Mike, and I'm very happy to present the research by Christian Dörig and Ludovic Chillier. They have generated an exciting data set and applied IPF to this. Okay, so the problem, Tina talked about this to quite some extent. The main assumption of most scoring algorithms is actually that there's one dominant peptide form per spectrum or peak group. And this is very often the case. We have, they elude at similar times these different positional isomers, but the algorithms, if there's enough evidence, they can actually differentiate nicely here. So here, this is the Q value, and you can see that the algorithm has no trouble differentiating between these two. But sometimes this gets more difficult, like when they're closely coeluting and they have shared transitions. You can see here that's a more tricky case where this is all kind of bleeding in. But in some cases, the algorithms are still able to differentiate. It depends a lot on the peak picking capabilities, but very often this works and the algorithms can differentiate between those cases. Sometimes it's very bad. Here, this is an example where this is perfectly coeluting. You can see it's a very closely related peptide forms, and the algorithm cannot differentiate. So what do we do in this situation? So IPF is very conservative. With the default parameters, it will just tell you, I'm not sure between these two things, and you need to do manual follow-up, because that's a good strategy when you're going to data sets with very large sample numbers. You don't want to focus on the things that are difficult. You want to get all the things that are easy to assess. Okay, so what they did to kind of characterize this problem a bit more is they generated a very great synthetic phosphopeptide data set. These are 530 cancer-associated synthetic phosphopeptides. They have 1 to 10 different phosphorylation sites and only one phosphorylation per peptide. So all these combinatorics, they get to about 1,500 phosphopeptides, and they're all synthesized. I mean, there's a large variation that we learned, but for our application, this is approximately equimolar in some cases. Okay, and then each phosphopeptide is measured twice. Once in a U-pool where all the isobaric forms are separated, so they're not measured together, and in an M-pool where they're all analyzed together. So we have a ground truth. We know what to expect, and that makes data analysis a lot easier. There's also low sample complexity. This was all measured in water without any background, and the pools were analyzed in DDA and SWOS, and they used the DDA library for SWOS. So on the first conclusion here is the retention time separation. This has come up a few times, and is actually an interesting topic. And they devised a strategy to assess this properly. It's very difficult if you have many different positional isomers and how you make these comparisons. They do pairwise comparison, and this reduces the problem to a much easier to quantify. So in all of these pairs, 72% of the pairs are perfectly separated. So this might actually be representative of biology because they're real peptides that would actually be measured. And 12% are partially separated, that might still be quantified by algorithms, and only 16% cannot be really separated. So the IPF performance on the U-pools can then now also be assessed. Christian analyzed all of this data manually, so there is a ground truth, and there's a lot of confidence that when something matches, it's actually a true match. And what he found is that IPF, the error model, actually performs accurately. So we have this loss of sensitivity compared to the human expert, because the expert knows the experimental design for this particular study. But in general, we retrieve the signals that we would actually expect. So we're always within the range of 60 to 90% of the phosphopeptides that we can manually detect and identify. Okay, and one important conclusion here was also that the performance is very dependent on intensity, and that whether you use neutral losses or no neutral losses for site localization, that also has a big influence. Now, interesting on the M-pools, what you can see here, immediately this large drop of sensitivity. The good news is the error model still holds through, even if there are many comparisons that do not lead to a success, but we just lose these site localizations because we cannot differentiate. And that's the default approach. Now, I say default approach because it's also possible to modify the IPF parameters of the model and to assess different combinations. That's something we're now looking into actively, and with the modified approach, we'll redo this analysis and see how we perform then and whether we can actually also better tackle coeluting positional isomers. Okay, so as mentioned, there are many different questions. There's also a problem of gas phase rearrangement and other challenges, and this will be addressed by a lot of upcoming papers. The focus of IPF is truly quantification across many samples and medium quality spectral libraries. If you're very interested in this, there are two algorithms I would like to point out that only focus on this deconvolution. One is Spectre from Ryan Peckner from the Jeffrey Lab. This is a very innovative approach, but it requires complete high-quality spectral libraries. You cannot have a single missing positional phosphopeptide isomer in this. And the other algorithm is Thesaurus from Baron Cyril from the McCoss lab. You already heard about this algorithm, and it really focuses on this positional isomer deconvolution. So check this out if you're interested in this particular problem. And as mentioned, IPF focuses on quantification across many samples. So to conclude the presentation, you have learned about all the challenges that we have between for PTM identification, and I would like to put re-emphasis on that most of these challenges are shared by the other mass spectrometric approaches, but we also have very nice advantages, and we believe that we can make use of these features with a series of tools. First, we need to do PTM identification. We need to build up a comprehensive library, and then what we can use is these PTM detection tools across many samples, and we're focusing on OpenSWOS and IPF, and this will enable us to draw biological conclusions from mass spectrometric data in a more consistent fashion. Okay, with this I would like to thank all my collaborators on this project, most importantly, of course, Udi Ebersold, who has been the supervisor of my PhD thesis, and all of the projects were conducted in his group with a set of very talented people. I'd like to also thank all the people of the IPF paper, especially Yansheng Liu, Hannes Röst, Tina Ludwig. This has been a very important collaboration, and I think the results will now apply this algorithm to many different data sets. For the coeluting phosphopeptides, I would like to thank Christian Dörig and Ludovic Chier, who conducted this study, and this will provide very interesting insights into this difficult problem. I would like to also thank my current group and the computational proteomics community, particularly the OpenMS teams, the TPP teams, the Skyline teams, for all these great collaborations on these projects. And thank you for your attention.
Terrific. Any questions about IPF at the moment? Okay, so I think we're going to go directly into the tutorial. Is there anything to say about that from your side?
Yeah, a few more slides. Okay, so just a few things, a few practical considerations if you want to apply IPF to your data. There's some things you need to think about. First, it's the experimental design. I would like to put emphasis on think about what's your technical validation. the hypothesis you're assessing and how you can validate this. So this is very important, especially if you're working with post-translation modifications, that you have a control on all of these different levels. Then when you plan your data acquisition, most important thing is really precursor isolation windows. You can use the best methods available, but really crucial for certain PTM types is that you have decent MS1 scans. So in the beginning, we only used 50 milliseconds MS1 acquisition time in this FOSS32 window acquisition scheme, and that turned out to be barely enough to enable such a study. So use the current schemes. If you have thermo instruments, that will actually be beneficial because we have seen that they perform a bit better for MS1 scans, and just make sure to use the best methods available. Then spectral library, that's also a crucial one. Eventually your PI might tell you, take the biggest phosphopeptide library anybody has ever created and apply it to your barely related sample. In this situation, you need to make sure that you're not doing this analysis because it will most likely not work. You really need a sample-specific library. You can use a repository library, but then please use mSplit DIA in advance to filter this down and make sure that IPF only gets an approximately matching library as input. The alternative is to do DTA library of representative runs. It always depends on your design. If you're interested in quantitatively assessing hundreds of samples, DI umpire is the way to go because this will build up your list in this dimension more or less good, and it will be a perfect representation of your DI data. And then you can do post-processing using site localization algorithms, but you don't have to. It depends a bit on the situation. If you do post-processing, you can be relatively less strict in terms of site localization. It's more important to kick the spectrum to the right site localization, because if you do a consensus, this can then slightly improve your peptide query parameters. Okay, then for IPF, you need to define the residue modifiability in the Unimod XML. We described this in large detail on the website. It's something you have to do either manually or you have to develop an algorithm for you to do it, depending on your research question. For the simple cases, we provide examples like phosphorylation. Then you generate the peptide query parameters. You extract and score the peak groups, precursor and identification transitions, and you do statistical scoring and inference with PyProfit. And what you then in this step need to ask yourself, do you need precursor inference on the MS1 and MS2 levels? We'll do it in the tutorial, so you see how it works. But if you do phospho analysis, you might not have to do it. It might be good enough to have tight precursor isolation windows, and the unmodified and the modified peptide forms will not fall into the same isolation window. Then semi-supervised learning. We usually now conduct this on all runs together. You will see this in the tutorial. And yeah, one important thing to also consider is how specific your peptide query parameters are. If you have a one-to-one matching between I have a peptide query parameter for this peptide form and I only care about this one, or you have very crude libraries where you say it should be approximately a peptide sequence with one phosphorylation, and you want to really score different peak groups, you can actually modify the statistical model. This will change a few of the assumptions, and it will enable you to make a much broader scan. So what you want to do depends a bit on your research question, and with these parameters you can tune IPF to get the best performance. If you don't care about anything of this, just use the defaults. It will provide reasonable results for many scenarios. And then troubleshooting. Eventually you will run into troubles. Always visualize data using Skyline or Spectronaut just to get a feeling for the data. One common issue, for example, is that, as I mentioned before, the library does not reflect the sample properly, and we have way too many poor peak groups. So with IPF, we expect to have rather this kind of peak group where we have a large set of transitions, like 5 or 6 detection transitions and several identification transitions. And not kind of this peak group here. This is borrowed here under something different. It only has 2 or 3 fragment ions, and IPF will not really like this. It will not be able to make anything useful with it because there are no identification transitions to extract. Now the good news is the approach is relatively robust. 1 to 10 ratio is not a problem. A 1 to 50 ratio is more difficult, and the 1 to 1000 ratio is almost impossible to deal with. The model usually calibrates itself, but in some scenarios it falls apart. So just make sure to think about what you actually want to do. And yeah, you can adjust this parameter here to automatically filter this a bit more, and then at one point it converges to this thresholding issue that I mentioned. Usually we set this very relaxed, but you can fine-tune this if you're going for very difficult applications. Important, getting help. Go to the website, read the instructions, and if you run into problems, contact us on the GitHub issue trackers or contact us directly by mail. So now we're doing the tutorial. This is the 6th part here. The good news is it's actually mostly the same as the OpenSWAS tutorial. All the steps are very similar. Here we use OpenSWAS assay generator with the IPF flags, we use OpenSWAS with the IPF flags, and we use PyProfit with the IPF flags. And all of it is done automatically for you, don't have to do anything. And the output looks exactly the same, just on peptidoform level. So what we're doing now is we actually have a little biological data set. This is based on this phosphoproteomic data set that we used for the comparison with DDA. But we're not extracting the full library. We're extracting sentinel protein assays. It's a concept from Martin Soste, from Paolo Picotti's lab. And we're using the particular implementation in the P100 assay set from Jacob Jeffy's lab. We use the protein IDs from there with the spectral library that's a bit fine-tuned. And it will enable us to actually figure out which parts of phosphopeptide signaling networks are actually affected by our 2 differential biological conditions, nocodazole treatment and controls. And then we will use MapTI to identify differentially abundant phosphopeptides and figure out what kind of biology is happening there. One important thing to consider now is all the steps are super fast except OpenSWAS workflow. The reason is we need to extract and score 300,000 transitions per run, and this will take more than the 5 minutes from the last tutorial. So skip this execution and directly copy the OSW files from the output folder and then directly continue with the PyProfit step to learn how to do this. Okay, that's it. Any questions?